Are we all ready to gather? Okay. I've got pigs, so. It's my pleasure to introduce Dr. Jonathan Trent. It's rare that a patient is able to introduce her doctor who she believes saved her life. After recovering from a ruptured cyst, sepsis, and finally surgical removal of my small bowel tumor, I sought a second opinion at MD Anderson. Dr. Trent immediately sought to get me in and keep me in the adjuvant trial for Gleevec, despite many setbacks. Here, almost 20 years later, I'm able to introduce him. After receiving his MD and his PhD from the University of Texas, MD Anderson Cancer Center, Dr. Trent became an assistant professor there in 2002. By the time I met him as a patient in early 2004, he had already begun producing articles, presenting at conferences, and serving on many GIST and sarcoma-related boards. Shortly thereafter, he became the founding faculty sponsor of our annual GIST Summit for GIST patients and their caregivers. In 2011, Dr. Trent was appointed Professor of Medicine and co-director of the Sylvester Cancer Center in Miami, Florida. He and his team focus on research and precision medicine for their patients, and his research has been used to devise treatments for patients based on their individual needs. I can attest to Dr. Trent's approach for specialized treatment, and I am truly evidence of it. Dr. Trent.
Thank you for the kind introduction. Honestly, it's a pleasure to be involved with the healthcare of somebody like Ginger, because she is active, involved in everything, and is just wonderful, amazing patient. And the energy is incredible. The GIST conference that she mentioned started with, I think, 13 people in the lobby of a condominium, and then grew to maybe like around 150 or something. And so it was one of the first patient educational conferences really for GIST patients. And so it was a real fun journey. So what I thought I'd do is talk a little bit about GIST, trying not to overlap with Robin too much. He gave us a lot of great information, and I know nothing about healthcare payers or anything like that, so I'll just plead the fifth. So we're talking about GIST, and again, we think about sarcomas, our pathologists doing what they look like under the microscope, and interestingly, GIST falls into this category of what you might consider the spectrum of neuromuscular tumors. There's features of nerve, features of muscle, and the precursor GIST cell, the interstitial cell of Cajal, really performs both functions and lives in that area. So it's a very different, even in the sarcoma, even though sarcoma is a very different cancer than carcinomas, GIST is very unique. It's also unique in that it's driven by a distinct point mutation, usually in KIT, but can be other. And then we're also really lucky over the years to have so many therapies become available, many sarcoma types don't have a treatment. There's no FDA-approved treatment for chondrosarcoma. Just was like that once, but now we have a lot of useful treatments. And we've heard today GIST is very common, the most common sarcoma, but what's also important is to understand that this is a mutation-driven tumor. It drives the phenotype, the mutations drive the therapy. It also has a high propensity to metastatic disease, even when there's a primary tumor. And so that's something that we screen for very closely. And that's where these tyrosine kinase inhibitors have been approved. And this is just a list of the tyrosine kinase inhibitors that have been approved in the US by the FDA. And we'll talk some about progression-free survival, or PFS, which is basically, it's the average time that the patient is on a medicine until their tumor starts growing again. So a median PFS of 24 months for imatinib, that's at the time point when half of the patients have had the tumor start growing again. The other half haven't had their tumor. There may be patients who I'm seeing now with metastatic GIST who had imatinib 20 years ago. And so it's just an indicator of activity. One of many, we talk about response rates and other endpoints, it's one of the many indicators of how effective a medicine is in a given situation. Now these numbers for sunitinib, rego, et cetera, are lower, but that's really because of the resistance to imatinib. We've been in situations where we've used ripretinib, sunitinib, rego in front line, and they're just as effective as imatinib in front line with a median of 24 months or longer. I would suspect maybe ripretinib might even be longer than that if it was used in front line, but we don't know the answer to that question. And so these are the common locations of mutations. We call these locations exons. And so the exons are really the location within the gene where the mutations occur. In GIST, it's most common in an area called exon 11 that's just inside the tumor cell. Think of the tumor cell as a sphere. Exon 11 is just inside of it. And that mutation is an autoinhibitory domain. So when you have a mutation in exon 11, you're removing the inhibition, and that kinase becomes active, just like turning on the light switch and leaving it on and taping it up, and nobody can turn it off. Exon 9 is a little different. It brings the two KIT molecules together, and then they activate each other when they're brought together automatically. It's just an automatic thing. Same with PDGF receptor. And then the others are rare. We talk about exon 13 and 17. Most commonly, we talk about them in the setting of resistance, because we'll get to it a little bit, those are the sites of resistance. But there can also be primary mutations in exon 13 and 17 that are sensitive to imatinib in front line. So there's a lot of nuances here with these exons, but that's basically how we talk about them. But then how does it function? So I mentioned that light switch. So this is what the light switch looks like when it's off. This is what it looks like when it's activated. So the light switch is on, it's sending a signal to the nucleus and telling now a normal cell to turn into a cancer cell, and these cells start splitting from 1 into 2, 2 into 4, 8, 16, 32, and then you've got a big tumor. And that's all driven by these mutations. And that it's unique in that it takes this one mutation in KIT, we think, to turn a normal cell into a cancer cell. And that leads to proliferation, survival, adhesion, invasion, metastasis, and all the bad things that the tumor cells do. In many of the carcinomas, like colon carcinoma, it's multi-step tumorigenesis. You have to have 5 mutations to get a tumor. Well, in this situation, it takes one, which is powerful, right? That's a powerful mutation, to have one mutation to turn a normal cell into a cancer cell. But it also creates a bit of an Achilles heel, because if we can block that mutation, then we can block multiple aspects of the tumor phenotype. And along comes imatinib, and imatinib displaces ATP from the binding pocket, and that turns the light switch off. And so now there's no electricity going to the light bulb's off. And all of these bad things that tumor cells do get shut down. And it's very, very effective. In KIT exon 11 mutated patients, the response rate is approximately 90%, if not a little bit higher. So it's very, very effective. And so that concept with imatinib also led to the regorafenib study that we've heard about, the sunitinib study that we've heard and know about, and more recently the Invictus study. And this looked at patients in fourth line and compared to placebo. We've heard a lot about placebo today. And Robin Jones showed this data showing that ripretinib is much better than placebo, and raised the questions of whether or not we should be doing placebo studies in GIST anymore at all. But this led to approval in the FDA, and then we've also heard about some of the barriers to approval globally. But the bottom line is that GIST is a molecular disease, and there's different GIST subtypes. Exon 11 and 9 we were talking about just now, but there's also PDGFR, SDH deficient. We have Ramesh Balusu here, who's a world expert on SDH deficient GIST and has built out an entire clinic based on this entity. The rare RAF patients, NF1, PI3 kinase. There's some evidence of IGF-1 receptor from Meg von Maren's group, and then recently described TRK fusions also occur in GIST. And these are all real things. And to know how to treat it, and then I mentioned the exon 13 and the exon 17 resistance mutations. These are becoming increasingly important. So 20 years ago, we were talking about GIST, and then we were talking about KIT mutated GIST. But all the GIST patients were treated the same. In fact, even recently, all of the GIST patients on trials like Invictus, trials like Intrigue and Voyager were just put on the trial as GIST patients and not taking into consideration the molecular subsets. But you can see that each one of these in white below are treated with different medicines than imatinib. Imatinib doesn't work in anything that's on white on this screen. So if a patient has a RAF mutated GIST, you wouldn't treat him with imatinib. You would treat him with something else. But you don't know RAF mutation is there unless you do the next generation sequencing. And a lot of people in this room I've worked with very closely over the years trying to raise awareness about mutation testing. It has to be done in GIST patients that are going to receive a therapy. The bad news in the US is that it's not being done. We looked at our SEER Medicare data up to 2015 and found that only 30% of metastatic GIST patients were having KIT mutation testing, meaning that 70% of patients were being treated blindly, which is embarrassing. This wouldn't happen in breast cancer. I don't know if people work in breast cancer or not, but breast cancer with HER2, you test for HER2 and you don't give Herceptin unless HER2's there. But that's not done in GIST, even today in the US. I think some countries around the world are much better about it, but in the US it's still a struggle. And I see these patients in clinic that haven't had testing. Not too long ago, I had a patient referred to me because of resistance to the first 3 lines of therapy. We did NGS and it was a PDGFR D842V. Patient shouldn't have had any of those treatments at all. Even the worst, Robin Jones talked about adjuvant testing. The same data showed also that even less than 30% of patients treated with adjuvant imatinib get mutation testing. And these patients are going on a medicine for 3 years that they don't need because they don't have the right mutation. And that's like, I think, probably $350,000, $400,000 in the US that's being wasted in a large number of patients. And so so the GIST community, our center, Lots of people around the world have taken this to the next level of awareness to start talking about circulating tumor DNA. Circulating tumor DNA is DNA that's in the blood. There's DNA in everybody's blood. Most of it is from normal cells, but circulating tumor DNA is DNA that's in a patient's blood that is being shed from the tumor. Tumors have their own blood supply. When tumors split from 1 into 2 to 2 into 4, there's one that dies there somewhere along the way. They have a finite lifespan, and they release their DNA into the blood. And it turns out the technology now has increased to the point that there are tests available to take blood from a patient, measure, collect the circulating tumor DNA, amplify it, and do the mutation testing on the blood. And so we do this now at Sylvester on any patient who's progressing on imatinib or any line of therapy beyond that. And I'll share with you some of our observations. So primary mutations are most commonly in KIT exon 11, KIT exon 9, and then other locations. And so this is data from about 170 patients, 162 patients from our center, where we found mutations. 106 had KIT or PDGFR mutation. Now then we also, I mentioned to you that that we do it beyond imatinib. That's because when a patient is newly diagnosed with GIST, we test the tumor. Okay, so we test the tumor because there's not resistance pressure from imatinib yet. So they're untreated, never had imatinib. You can biopsy a nodule, you can collect DNA from a paraffin-embedded tissue, and you can do the testing on the tissue. You don't need circulating, we don't typically need circulating tumor DNA at that point in time for the way we manage our patients. But when the patients become resistant, we do circulating tumor DNA testing because different tumor nodules can have different mutations, and we want to know what is the dominant mutation in the blood, which is a parallel to the dominant mutation in the tumor or tumors. And so what we found in terms of resistance mutations is that there's 2 types, exon 13, exon 17. They do different things to push imatinib off of the KIT molecule. So it can't bind to it anymore, and it can't keep the light switch on. It can't keep the light switch off. Imatinib can't keep the light switch off with these mutations. Either 13 or 17, the light switch comes back on. Most commonly, we find them in almost 2/3 of the cases in exon 17. That's been shown in 2 or 3 studies now, so it's been verified. Other mutations show up, but exon 17 and 13 are the most common. Now, what are exon 13 and 17? Well, one of my colleagues when I was at MD Anderson, Lei Chen, was the first to publish these resistance mutations. They were observed in leukemia, treated with imatinib, but what she looked at was patients who had multiple nodules and had those nodules resected and found that exon 13 is the ATP binding pocket, and exon 17 is this loop that can come up and block imatinib from getting into the ATP binding pocket. So those are the 2 resistance mutations that we'll be talking about, 13 and 17. Why are 13 and 17 important? Why are we talking about exons? Well, there's for a number of years now, there's been emerging data from Cesar Serrano, who's going to be here tomorrow, and we'll talk about this in a lot more depth and a lot more science. But his lab, some of our observations have found that exon 13 and 17 resistance mutations are associated with differential sensitivity to subsequent medicines. So for instance, if you look at this heat map, green means sensitive, means imatinib kills the cell. Red means resistant. Yellow means somewhere in between, or we don't know yet. And so you can see that all of these medicines down the left side that we've been talking about, these tyrosine kinase inhibitors, they all work really well at primary mutations in exon 11 and exon 9. But then you start looking at the resistance mutations and you start seeing some differences. The major difference today is exon 13. This may be different tomorrow. This may be different after THE and IDRX that you heard about. But as of today, sunitinib is really the only one that's consistently and potently green at exon 13 and 14. Mostly exon 13. Exon 14 is a rarer mutation in the same area. But the other ones aren't. You can look at regorafenib, the bezuclastinib, Pexi, ponatinib, avapritinib, ripretinib. They're not that active at exon 13. In fact, we've seen that when we thought they were, they're not. Sunitinib is the most active by far. And then if you look at exon 17 and 18, sunitinib's not very active there, right? So, but the others are. Most of the others down the far column on the left are active at 17 and 18. So that's made us think, why are we treating patients that progress on imatinib? Why are we treating patients who progress on imatinib with sunitinib, then regorafenib, then ripretinib, because that's the order that we did the studies? Does that make clinical sense? Does that make biological sense? Or is it just an arbitrary timeline of when they were developed? So we decided maybe we should put some more thought into it and study it a little more. And so I'll share with you some of the lessons we learned over the years. So this is a 52-year-old woman with a small intestine GIST, KIT exon 11 mutation, liver metastases, responded to imatinib for about 2 years, progressed on imatinib, was placed on the KIT inhibitor avapritinib. And this was years ago, and had rapid progression. When she progressed on imatinib, we sent the circulating tumor DNA out for testing. This was fairly early, and it took about 3 weeks to get the test back. And so she ended up progressing rapidly. You can see from the baseline to the middle panel, this was in about 3 weeks, and she ended up in the hospital. And I went to see her in the hospital with the circulating tumor DNA results and found that it was exon 13. And that explained why avapritinib wasn't doing anything, because it has no activity against 13. And so it's not easy to do in the US to get sunitinib in the hospital, because these drugs are expensive and the hospitals don't want to carry them. But we made a big deal about it. We got the sunitinib. We had to get consent from the patient's husband because she was getting confused from all of the liver involvement. So we got consent from the husband, and we started giving her sunitinib through a G-tube, and she had an amazing response. She had exon 13 treated with sunitinib, amazing response. Great. You can see how dark the tumor is on the right. It is a little bit smaller, but importantly, look how dark it is. This is a sign of a great response. And so to me, seeing this happen to a patient is really inspiring. And I knew that there's a future in ctDNA, and we've got to get this ready for prime time. So that's one of the initiatives at Sylvester. And here's another patient. This patient actually called me after being recommended to go to hospice. He said, Dr. Trent, is there anything else I can do? And I said, well, let's do circulating tumor DNA and see what you have. And so we did circulating tumor DNA and found exon 17 resistance. The patient had been on 5 lines of prior therapy, but had this exon 17 resistance, had already had regorafenib, had already had, ripretinib wasn't available, but had regorafenib, sunitinib, imatinib, nilotinib on a clinical trial, and something else. But then we decided to get off-label ponatinib because it had been described by Mike Heinrich and some others to have really high activity against exon 17 resistance mutations. So we put him on ponatinib. And on the left panel, you can see bulky, large tumor with the dotted lines going across there in the liver. And then you can see them getting smaller and, importantly, getting a lot darker. So this was another good response to therapy that was based on circulating tumor DNA. So we decided, since we started doing it regularly for those two reasons, we decided to take a look at one point a little while back, look at our experience with our patients. We published this in JCO Precision Oncology in 2020, if anybody wants to look at it. We looked at 243 GIST patients that had circulating tumor DNA. And then we looked at different groups of patients you can see on this diagram. And then we delved deep into our patients at Sylvester. And we found that there was concordance. If a patient has a primary mutation from an archival specimen from a surgery 5 years ago, when we do circulating tumor DNA, there's 100% concordance with the primary mutation. It may not replace the FFPE, but if a patient is having a biopsy and we're having a hard time getting tissue biopsy, we'll do circulating tumor DNA because it's, based on our data, it's trustworthy. The other thing that we learned is that small numbers of patients, but when patients had a primary GIST, not metastatic, and we did circulating tumor DNA, we didn't detect any mutations. So primary GIST should really have a biopsy. The other thing that we found is that patients had low volume disease, were on an effective TKI and not progressing. You almost never get any circulating tumor DNA. It's only the patients who have progressive disease, have larger volume metastases, that you can detect circulating tumor DNA. In our study, we detected it 100% of the time with those parameters in place. So next, we just decided to look at the outcome. So at our center, we compared patients that had ctDNA testing and looked at their progression-free survival and overall survival. And these are patients that have multiple lines of therapy. This isn't frontline therapy, this isn't second-line therapy, this is multiple lines of therapy. And so what we found is that the median progression-free survival was just over 6 months. And so these are patients who've had sunitinib, who've had regorafenib, who've had imatinib, may have had some other agents, and then we did circulating tumor DNA testing. And if they had 13, we put them on sunitinib. If they had 17, we put them on rego, and found when we do that, the median PFS overall is 6 months, which is just as good as Sutent in second line. Normally, as you saw earlier, the progression-free survival gets lower and lower over time, but we're able to elevate that by implementing circulating tumor DNA. And so that led to some other analyses. So we begged and bartered and worked with Blueprint, and Blueprint was very willing to work with us on this. And this was largely driven by Cesar Serrano. He'll talk about it in more detail tomorrow. But in the Voyager study, they gave us, we all participated in it, the GIST community, and put patients on the study, and we asked them to give us the data from our patients back for us to look at. And so they did give us the circulating tumor DNA data on a large number of patients, 386 patients. And so what we found in the Voyager trial is if a patient has a KIT exon 13 resistance mutation and are treated with regorafenib on the red curve, they do much better. Higher up to the right is a better outcome. Lower down to the left is a worse outcome. So you can see how far apart those curves are. And that's a matter of a large percentage or a large number of months. So patients with KIT exon 13 benefit much more from regorafenib than they do from avapritinib. And then conversely, when we looked at patients who did not have the KIT exon 13 mutation, so in patients who don't have exon 13 resistance mutation, they remain progression-free on avapritinib compared to regorafenib. So this is no exon 13. So if there's no exon 13, that means they have 17, and 17 benefits from avapritinib. It was one of the ones at the bottom of the chart on the left that's very active at KIT exon 17. Now, it's not approved in the US for that indication. It's for PDGFR 18, but it's very active at KIT exon 17, and sunitinib is not quite as active at that location. So time passes, we get more patients tested, we get more information, and so we decide to look again, and we presented this, Steve Bialik, one of our fellows, presented this at CTOS this last November, and I'm excited to say that he signed the contract and joining us as faculty at Sylvester. We're very excited. He's going to be very busy, though. And so we looked at the KIT alterations, and again, about 2/3 of them are in exon 17 and 1/3 in exon 13. And then again, we looked at one of these Kaplan-Meier's to look progression-free survival, and this was amazing to us. So we looked at our patients with KIT exon 13 resistance mutations, and if they're treated with sunitinib, their time to treatment failure was 9.6 months. If they're treated with regorafenib, it's 4.8 months. So circulating tumor DNA testing doubles the outcome. Patients do twice as well if you choose the treatment based on the circulating tumor DNA. And this was statistically significant. Other included Ripretinib and Avapritinib and some other drugs. And we're going to be very excited to look at this data again so that we can understand the results from the newer generation of inhibitors. So there's other mutations, though. We talked about exon 9. Exon 9 is on the outside, and it makes the receptor sticky. The kinases inhibit on the inside, so you have to use higher doses of imatinib. So from the left panel to the middle panel, there was progression on 400. We did mutation testing and found it was an exon 9. We escalated the dose to 800, and the patient had a great response. Same with RAF. This is a patient that I've been seeing on and off for 20 years now. I guess about 10 years now. Probably about 15 years I've been seeing him on and off. But he ended up having a RAF mutated GIST, was treated with some other things, some KIT inhibitors that didn't work, found the RAF mutation, got him actually on a clinical trial with a RAF inhibitor that was new at the time, and he had a good response to therapy. And then this is another example of D842V. You heard about this from Dr. Jones, and you can see that Avapritinib is the drug of choice for D842V because almost everybody gets some tumor shrinkage from it. There's some caveats that he mentioned with the side effects that we've got to be careful with, but you have to do the mutation testing. I tell the community oncologists, and not surprisingly, maybe surprisingly, maybe not, but some sarcoma centers don't do mutation testing regularly. It's mind-boggling, but this is one of the reasons you have to do it, because these patients benefit so well from this therapy. So this is a little bit of the roadmap that we use. We find the mutation, we identify the inhibitor, we draw the lines together until the patient, with the NGS, the patients get optimal therapy based on NGS testing. And then we heard something about the ongoing clinical trials, and we have the phase 1 IDRX, THE630, NAB003. These are all newer generation KIT inhibitors. We don't know what the results are going to be. We don't know if they're going to be better than Ripretinib or not. In the lab, they are. We don't know what the side effects are going to be. We're figuring all of that out right now in the clinical trials. But it's good to know that patients with metastatic GIST have these options. And most of our centers have all three of the studies open. So if one of these doesn't work for the patient, we'll get them on the other one quickly so that they have a better chance. And then I'd also like to put a plug in for our ctDNA study. This is a phase 2 study where a patient with progressive metastatic GIST has a tumor growing. We do ctDNA testing. If they've got 13, they get sunitinib. If they've got 17, they get regorafenib. Doesn't matter what line of therapy. They could have had 10 different agents by now, but if exon 13 is driving that resistance, we want to see how sunitinib performs, and even if it's a re-challenge. And this is largely for patients that might not have other treatment options. We'll re-challenge based on the ctDNA testing. And then the phase 3 studies that are open are the Cogent study, which is sunitinib to target exon 13 by itself, or sunitinib plus bezuclastinib. Bezu targets exon 17. So exon 13 and 17 are both being targeted by this combination. We think THE covers both. We don't know yet, but we already do know that Sutent's very good at 13 and Bezu's very good at 17, so this phase 3 registration study is looking at the combination. We'll be presenting an update of the phase 1 study at ASCO this year. If anybody's interested, this bezuclastinib plus Sutent versus Sutent alone, we're going to be presenting that updated phase 1 study results at ASCO in a few weeks. And then there's the new study that we spent a lot of time talking about in controversy with Dr. Jones, the Ripretinib versus Sunitinib in 17-18 resistance. So I'd like to thank the GIST team back at Sylvester and the GIST and sarcoma team back at Sylvester, for which none of this research would really be possible, or I couldn't get away and be here with you if we didn't have a great team taking care of the patients. And I'd like to thank you, everybody, for inviting me to speak about this really important topic that we've been at Sylvester and in my career have been passionate about finding a cure for many years. We're getting closer, but we got to keep working. Thank you, and I'd be happy to have any discussion or answer any questions.
Ripretinib showed some activity with 17. How come they didn't re-explore exon 17 with that drug?
That's a good question. I think there was some discussion. I don't know anything officially, but I did hear some discussion about that as a possibility. That was proposed by some of my colleagues. Also, combining avapritinib with sunitinib could be an approach also. But I think that with some of the toxicities, I think the company is, they're very good at developing drugs, and they develop them very quickly with their platform. So I would suspect that they're probably developing even better just medicines that are going to have fewer side effects is what they're focusing on.
John,
Yeah.
Thank you for your fantastic presentation. Just a question to Avapritinib. Because of these cognitive side effects, are you using any kind of tool to assess the situation of the patient, maybe from the neurology colleagues, to see what is the situation of the patient in the direction of quality of life, where you can really track the patient, because sometimes maybe patients don't have relatives. So if there is a cognitive side effect, maybe the patient is not aware of it, but the relative is aware of the side effect or of the cognitive deficit. But if someone is living on his own, I think it's quite problematic.
Yeah, it's two very good points. Two very good points that, with the cognitive toxicity from avapritinib, just like other types of, it's similar to a dementia, patients are very good at covering it up. They're very good at covering it up. And oftentimes it's somebody else that lives with them that tells us that they're having these side effects. And so that's one good point. The other point about neurocognitive testing, we don't do it as baseline in everybody. Maybe we should. You're making me think maybe we should do it in baseline with everybody. We do it in older people who may have some pre-existing dementia or vascular disease. We do baseline in those situations. And if they do start showing signs, we do, or the caregiver tells us, we do neurocognitive testing. And it's very specific. What's missing is very specific and is consistent with drug association in the setting. And so, but then also importantly, the neurocognitive deficits that are identified, the neurocognitive psychologists and counselors can train them how to overcome the deficits by making lists and some other tricks to help them manage the neurotoxicity, because as you saw, there's a good chance the drug's going to help them. And the neurotoxicity can be scary, so we have to really balance this and try to minimize the toxicity so that we can treat the tumor. But very good points. Dr. Belusu.
John, thank you very much indeed. It was a fantastic talk. This circulating tumor DNA kit you're using in your hospital, is it in-house or are you using commercial kit?
We are sending it out. It's a send-out lab for us.
Right. And has there been any issues with regards to quality assurance, that kind of thing, or is already
Yeah.
been dealt with?
Yeah, so it gets billed to insurance. If insurance doesn't pay, then the company ends up not charging anybody. Because like the NGS, they're selling the data to a pharmaceutical company, so they're making money anyway. If a patient gets a bill, I ask them to give me the bill, and then I call up the company and I say, look, you can't do this, or we're not going to order your test. And so they don't charge the patients.
Thank you.
I don't know if that's good or bad, but it is what it is.
Hi, John.
Dr. Jones had a question? Oh,
No, you go ahead.
Hi, John. Thanks for your presentation. Quick question. How effective is 800 milligrams of imatinib for a patient who has an exon 11 mutation who has progressed on 400 milligrams?
yeah, it definitely has some activity. And before sunitinib became available, that was an option. When sunitinib became available for renal cell, we started using it, but 800 was studied in the Korean group, Yoon-Kun Kang, and they showed that it was a median progression-free survival of about 3 months. So better than a placebo, better than an ineffective therapy, not quite as good as an effective therapy, but definitely beneficial. In fact, there's been some dimensional responses have been reported in exon 11. We tend to use it as we're bridging a patient to a clinical trial. So if a patient is progressing on imatinib and we want to evaluate them for the bezuclastinib plus sunitinib trial, then we'll ask them to escalate their imatinib to 800 just to make sure that their tumor's not growing. We can stabilize it some until we get them on the trial. Oh, I think Dr. Jones was next, and then we'll get you.
So, just a comment, and I'd value what everybody thinks about this, and it follows on from a comment that Marcus made in the last session about patients with just being treated outside of reference centers. What are we going to do in terms of, for instance, as you said, the mutational testing in the United States? It's really shocking every time that's mentioned, and things are going to get more sophisticated and more tailored, as Marcus mentioned. So, what do you think of that, John? And how is that going to be improved? Getting the message out to community oncologists in terms of the need to actually be precise and thoughtful in terms of managing this condition, because I think sometimes it's felt, oh, this is straightforward. We just need to start imatinib and sunitinib, regorafenib, etc. So, what are your thoughts? I mean, it's more of a sort of philosophical question than anything else, and there's no direct answer. But.
No, I think it's a real question, and it's a real critical issue. We've been taking care of patients with rare diseases for many, many years, and it's hard to reach the community oncologists in terms of even the right treatments. We in the U.S. we still see some crazy, you see nilotinib given in second line and things like that. So we just have to keep hammering home education, publications, review articles, presentations, CME events, and working with the advocacy groups who are in touch at the ground level with the patients. We just have to keep keep working in as many different directions as we can. And then another answer is clinical trials. You know, you were just telling me we need to do these clinical trials because it gets access. We need to be doing clinical trials with ctDNA. And that's one of the good things that I see out of the Ripretinib versus sunitinib exon 17. If that's a positive trial, that's going to validate that patients need to have, prospectively, patients need to have ctDNA testing. And that's why we opened our trial, is we want to have prospective data that shows that it improves patients' outcomes objectively, rather than me showing a bunch of cases where we think it's effective. So I think we have to keep doing the research. We have to keep working with the companies. And, you know, hopefully the regulatory agencies will realize that this technology is actually improving outcomes and saving money. I mean, you would think the ctDNA testing would save money because you're not treating patients with, you're more likely to treat a patient with an effective therapy than an ineffective therapy.
Absolutely, and I think the INSIGHT trial in that respect is really, really important in terms of getting that message out there almost. Yeah.
Yeah, good question. Yeah. You want to ask Robin a question? I'm fine with that.
Because we have one of our colleagues here is a pilot, and this is Jörg. He was a pilot, and we have had a discussion that as a pilot, if you try to fly a new model, you have to get a training. But as an oncologist, it seems that if a new drug is available, everyone can do it. So the question is there a chance maybe really to come up with a limitation so that you really work with the companies and say, okay, we are thinking that we need to do a better work within the country, and so we offer a specific training, and it's more or less a must. If you want to prescribe the drug, then you need to have this specific training.
Yeah.
I don't know.
I mean, I think it's you know, I think there's advantages to that. It makes sense. We do, you know, there's lots of things in our hospital that become new, and we have to do training. It's required. And, you know, I think that's not a bad idea. When we do a clinical trial, we have to train all the physicians on the drug because it's a new drug they're not experienced with. But when a drug is FDA approved, you're right, there's not universal training. Even in our hospital, I mean, most of us at our center, we've been involved in the trials, but you know, I imagine at many centers for the common diseases, they're not, and in the community, they're not involved in the trials. The drug gets approved and they don't know what they're doing and just start prescribing it. I don't know how to implement something like that. It would be very difficult in the U.S. I don't know. Yeah. Yeah. That's true. That's true.
It's it's such a contradiction in many ways that for a clinical trial we have to do so much in terms of training, but then when a drug is approved, oncologists can just use it.
Yeah.
So we learned quite early on that among the KIT mutant GISTs, that exon 11 and exon 9 were really very different. But we were looking at some papers recently in the science group that even within exon 11 category, there's really a very wide spectrum of different mutations that occurs. Is the evidence, do you think, beginning to accumulate that they're not all created equally? That there are some distinctions among the different exon 11 mutations, or is that a reach?
There's some, you know, we all thought there would be a lot bigger differences than there are. There are some differences, but they're not that large. 578, 579 is associated with possibly the fastest growing GISTs. It really it's associated with the highest mitotic rate. So there are some differences in that. Whether or not it impacts other resistance or even survival, I'm not aware of any robust evidence.
Is anybody looking for it?
Yeah, people have looked. Yeah. Okay. Yeah. Maybe Robin or Ramesh know something about that, but I'm not really aware that the deletion, larger deletions or point mutations really have much of a difference in ultimate outcome or drug sensitivity.
I just wanted to have a quick question on what you said made absolute sense to me, which is that you'd think that it's more cost effective to have an effective drug treatment, which you've figured out through the ctDNA, than to have an ineffective one and keep somebody on a drug that's costing a lot of money for months. Is there like a set of guidelines in the US where you say, if there's a GIST patient, then you should follow these steps, and the ctDNA is one of those steps, when you get to a certain level of maybe imatinib stop working, etc. So you insert there and say, yes, now you must have ctDNA testing done. Do they exist? And if they exist, well, okay, so they don't exist.
No, in fact, most centers in the U.S., I don't think most centers in the U.S. routinely do ctDNA testing. I think we're probably the major center. So the major ctDNA company in the U.S. is Guardant 360, and last time we got all of their GIST data, ours were half of their patients. So I think that our center is doing the majority of the testing. I think some other centers are doing some, maybe starting to do more, but it's not routinely done in the U.S., and it's not on our guidelines, our National Cancer Center Network guidelines. This may or may not surprise you also. Our National Cancer Center Network guidelines don't even require KIT mutation testing or any mutation testing in the U.S. if you're going to treat somebody with imatinib. It's recommended, which to me, again, is just unbelievable. And so there's no mention of ctDNA in the NCCN guidelines. We've tried, but there's certain individuals and centers that don't think that we should be that dogmatic. Sarah, I've been a part of that with Sarah. Maybe Sarah wants to say something.
I think you've summarized it well. We've definitely tried to get on the NCCN committee as a patient advocate, just to also express our voice, but they're also very particular about who's on the committee. But even among the committee members, it's been very difficult in terms, they aren't willing to take the issue to the team. It has to go through a process where you submit a letter and then it has to wait a year until they review it. And it's unfortunate that that's what's taking place. Because the thought is, just addressing what Robin's saying, how do you get mutational testing done by community oncologists? The thought was NCCN would be a good start to have it required. So if they look that up, they know that's part of the formula, what they need to do.
Yeah. So it's basically like
patient community.
Yeah.
When the patients contact us, we tell them to go to a center of excellence that will do the testing.
Yeah. So to your point, the trials were done in a certain order. Imatinib was in front line. When patients became resistant to imatinib, there was the sunitinib trial. And then when patients became resistant to that, there was the regorafenib trial and then the ripretinib trial. And so that's the way most centers dose the agents in that chronological order. But a lot of us are questioning that.
The World Health Organization, for example, that could impose mutational analysis testing on all these countries' guidelines, for example.
And it gets tricky globally because there's a lot of countries that don't have the technology. So it gets tricky globally, and I think we discussed that also with LifeRaft Group and the LifeRaft Group's own language about requiring mutation testing. A number of people were against it because there's countries that in some areas in the world that can't do it. And so we don't want to say that it's required when we realize that there's some people that can't do it. So it's, it's, to me, initially sounded pretty straightforward, but there's a little more nuance to it than I expected. Ramesh?
John, hi. It's a comment from Basically, what is happening with the KIT exon 11, the spectrum of mutations, is there's an excellent paper from Lorena and Bruno from Italy. So Lorena is from Palermo and Bruno is from Rome. They are looking at the topography of the mutations in the exon 11 from 550 to 584.
Yeah.
And there's an interesting data set. It's retrospective, so it's not in a big randomized series or a prospective series. 557, 558 seems to be deleterious.
Yeah.
We all know that, and we've been known for a long time.
Yeah.
What is interesting is if your mutation in the exon level is more towards 570s, 574, they tend to have less aggressive cores from the data set. Same thing if you're about 550, 555, and again 560. So 557, 558 seems to be a red flag.
Yeah. I think I misspoke. I said 578, but I meant to say 558. That's the hot spot that's associated.
557, 558 seems to be the red flag.
Yeah, that's the highest mitotic rate.
Another thing Heike and myself we are thinking about is whether to incorporate the mutational analysis data into risk stratification.
Yeah.
Because if you look at the risk stratification, it says mitotic index, tumor size and tumor site. But we know now that the mutational analysis, especially for the KIT exon 11, has an influence on the PFS. So perhaps we should be looking at that in the next time when we look at the stratification model, we should put the mutational analysis into the pot as well.
Yeah, that's a good point. And we know some GIST like SDH and even PDGFR behave differently than KIT exon 11 558, and the risk of recurrence is much different. Any other questions? Okay, thank you.